telling. If the sugars are alpha glucose units, you will call it as alpha glycosidic bond, and you will see which are the carbon atoms linked. So the linkage in case of maltose is known as alpha 1,4 glycosidic bond. Okay, alpha 1,4 glycosidic bond. Okay, now tell me what is the linkage in lactose known as?
Beta
Beta.
linkage.
What is the number of carbon? So how will you tell? Beta. This was alpha 1,4 glycosidic bond. So what will be this known as? Beta.
Beta 1,4, madam.
Beta
The
1,4
one four
glycosidic
1,
bond. Okay, what is this linkage known as? Alpha
1, 2.
1
one
to
two
glycosidic bond.
2
Okay, so because
glycosidic.
1 and 2. So from the glycosidic bond itself, you will get an idea whether the sugars are existing in the alpha or beta conformation and between which carbon atom the linkage is mediated. Okay. Now coming to the important properties of this monosaccharides and disaccharides reducing sugar. So this is very important, which you'll be doing in the qualitative analysis today. So reducing sugars. All monosaccharides are reducing sugars and few disaccharides are reducing sugar. Now what is a reducing sugar? A reducing sugar is one which can carry out a reduction reaction. And when can they carry out a reduction reaction? If they have a free aldehyde or a ketone group. Okay, consider the monosaccharides. All of them, even in the ring structure, they will have this free hemiacetal group. Okay, so the O and the anomeric carbon, this forms the hemiacetal group. In monosaccharides, it is free. Even in the linear form, CHO is free. But when they form a disaccharide, in case of maltose and lactose, the acetal group is free in this case. Okay, see this acetal group is free, whereas this is involved in a linkage. And here this acetal group is free, whereas the other is engaged in a linkage. So maltose and lactose has a free acetal group. So this is a reducing sugar. But when you come to the sucrose, they doesn't have a free acetal group. Okay, I'll show you another. So see, in case of lactose and maltose, the acetal group is free, hemiacetal groups are free. But one is involved in the linkage which is not free. But take the structure of sucrose, both the acetal groups are involved in a linkage here. It does not have a free acetal group as compared to the maltose or lactose. So all monosaccharides have free aldehyde or ketone group. So all the monosaccharides are reducing sugars. When you come to disaccharides, maltose and lactose has free hemiacetal group, so they are reducing sugars, whereas sucrose is non-reducing sugar because it has the acetal group locked in the linkage. It does not have a free hemiacetal group. Understood? So why all monosaccharides are reducing sugars? Why all monosaccharides are reducing sugars?
Because they have the free
because they
Because they
they
have a
have
free
free
acetyl
aldehyde
group.
or
Yeah, good. They have
acetal.
free aldehyde. Okay, what are the disaccharides which are reducing sugars? What are the two saccharides that are reducing sugars?
Maltose and lactose.
lactose and
Maltose and
maltose.
Yeah.
lactose.
Sucrose is a non-reducing. This kindly remember, it is a very important test to identify which is the unknown sugar given to you practically. Okay. Now we'll move on to polysaccharides. So polysaccharides are more than 10 carbon units linked together. So see here, so it has n number of glucose units linked together. See, polysaccharide, multiple glucose units linked together, they form a polymeric structure. So you have different polysaccharides. If the polysaccharides are made of just one type of monosaccharides. They are known as Homo Polysaccharides. For example, the starch and cellulose, they are made up of only glucose, no other monosaccharides. Okay, but heteropolysaccharides are those which has repeating units of two monosaccharides. Like heparin, it will have two different monosaccharide units forming a polysaccharide structure. Okay, so homopolysaccharide, all the sugar units are same. In heteropolysaccharide, they do not contain same sugar units. Different sugar units are together. Okay, now let us study the properties of few polysaccharides: cellulose, starch, and glycogen. Okay, cellulose is found in plant, starch is also found in plant, glycogen is a polysaccharide found in animal. Okay, cellulose function is it is present in the cell wall. Okay, you can see the cell wall, they've enlarged here. A cell, microscopic structure showing the cell. The wall contains this cellulose, which is made up of beta glucose units. Okay, so cellulose is made up of beta glucose units. What is the linkage? Beta 1,4 linkage, and they are linear structure, no branching. They just form a straight chain. This is the property of cellulose. When we talk about starch, it functions in plant as a storage polysaccharide. Glucose will be stored as starch. In animals, excess glucose will be stored as glycogen. They are made up of alpha glucose units. Cellulose is made up of beta glucose, whereas starch and glycogen are all made up of alpha glucose. Now you see that the starch has two parts, amylose and amylopectin. Amylose is a linear polysaccharide, so all glucose units are linked by alpha 1,4 glycosidic linkage. Whereas amylopectin is a branched structure. Okay, see here, this is a linear structure, a straight chain just folding. But when you take the amylopectin, it is branching. See, it's a linear structure now and it starts branching. Linear, it starts branching. Linear, branching. So it is a branched structure. So wherever the molecules are linked linearly, the bond is alpha 1,4 glycosidic bond. When there is a branching, it has alpha 1,6 glycosidic bond. Okay. When they are linked linearly, the bond is alpha 1,4 linkage. But when they branch like this, between first and sixth carbon atom. So the branching position will have a linkage which is alpha 1,6 glycosidic linkage. So this is present only in amylopectin and glycogen because they are branched molecule. You can observe here. Whereas this is a linear molecule, so you will have just 1,4 linkage. Wherever branching occurs, there is 1,6 linkage. And now, in amylopectin, branching occurs after 20 amino acids, 20 glucose units. Whereas in glycogen, branching will occur after every 10 glucose units linked together. Okay, is this clear now, the polysaccharides? Okay, so this I just gave a basics to what are the different. Now can you tell me, will the polysaccharides act as a reducing sugar? Can they act as a reducing sugar? They are highly linked molecules and they are all branching molecules. Will they have the free aldehyde or the ketone group?
No.
So they are
No,
non-reducing.
madam.
So polysaccharides are non-reducing. Okay, whereas monosaccharides are all reducing. In disaccharides, lactose and maltose are reducing. Sucrose is non-reducing. Okay, so we've completed the structure and properties of the carbohydrates. What are the different classes of carbohydrates? So now practically what is important to us is we'll give you a sugar sample. If you had come to our lab, you would have had this experience. We will give you a sample, either glucose or fructose or maltose or lactose, starch, whatever, we'll give you a solution and we'll ask you to find out what is the carbohydrate present in the solution. That is your task for the practical component. So I told you all monosaccharides are soluble. They will not make any difference. Okay, and even they'll be sweet to taste, so you cannot identify them. You have to do certain tests and then identify whether the unknown given to you is a glucose or a fructose or galactose, or whether it's a maltose or a lactose, or whether it's a starch solution. You have to do certain tests and you have to identify. Now, what are the tests which you can do for carbohydrates? So, all of you, please listen carefully from this. Try to understand anything you don't understand. You can stop me. What are the different tests which you can do for carbohydrates? So, can you tell me what are qualitative tests and how do you do qualitative tests? What do you mean by qualitative tests?
Where we find out what is the compound or substances present into the
Yeah.
solution, not the
You're
amount.
finding out
Yes.
what is present. And one more thing I told you, the reagents and the volumes need not be accurate. So here you will not measure, you will approximately take some volume and then you add everything and check for the test results, whether it's positive or negative. Okay, so these are the qualitative tests which you will do for carbohydrates to identify which sugar, or whether it's a disaccharide, whether it's a polysaccharide, you can identify by doing certain tests. Now, firstly, I'll discuss what are the different tests which you can do, and then I'll tell you how you can identify which is the sugar present. So, first, let's discuss the test. So, first test which you can do for the carbohydrates is solubility test. Okay, you will take the sugars, dissolve it in water. Okay, when you do this, you can do for the carbohydrates, you will take around 10 ml of water. Okay, you will take a test tube and you will take water in it, roughly cold water, and then you will add the sugar to it. Okay, a small pinch of sugar to it, and you will dissolve it, mix it. So when it's dissolving completely, it says that it's monosaccharides or disaccharides. Okay, either monosaccharide or disaccharides, they are soluble in water. So they will form a clear solution. Whereas if you dissolve starch in water, have you all observed how will it look? Will it form a clear solution? If you
No,
dissolve a
ma'am,
starch in water,
Turbid.
it will be
it will be
cloudy
turbid.
solution.
Turbid.
Yeah, it
Turbid.
will be
Cloudy
turbid.
and turbid.
Yes, so this is a polysaccharide. So this to an extent will tell you. So what you can make out from this test, whether it's a polysaccharide or not, but you cannot distinguish between monosaccharide and disaccharide. If it is completely dissolving, you can tell it can be monosaccharide or it can be a disaccharide. If it's not soluble and it forms a turbid solution, you can say it is a polysaccharide. Only that you can determine. So what is the next test you do for carbohydrates? Maulish test. This is in general for all the carbohydrates. All the carbohydrates will answer the test. You can say whether a carbohydrate is present or not. You cannot tell whether a monosaccharide or a disaccharide or a polysaccharide is present. You can tell whether carbohydrates is present or absent. So what do you do in case of Maulish test is, you will take the sugar solution, any sugar, you can dissolve glucose, fructose, lactose, maltose, starch, whatever you dissolve in water, you take the carbohydrate solution, okay, and to that you will add Maulish reagent. I'll show you the video also. Before the video you have to understand the concepts, no? So I'll explain now and show you the video, you'll understand better. So you will take the carbohydrate solution, you will add Maulish reagent to that, mix it well, and then you will hold the test tube in a slanting position and add H2SO4 to the sides of the test tube, not to the solution. Without disturbing the solution, you have to add H2SO4 drop by drop to the walls of the test tube. And what will you observe is a brown ring or a violet ring. Okay, so I'll show you the picture. Observe here. So this is a blank where there's no carbohydrate, negative test. This is a positive test. See here, brown or a violet ring will be formed in between two layers. Okay, so this is a negative test. This is a positive test. So what is the inference from this test? If it is a negative test, what will you tell?
The sugar is not present in the
Yeah.
Carbohydrates are
No,
absent.
absent.
or
Okay, what will you tell
carbohydrates are
if you get a positive test?
Carbohydrate
carbohydrate
is present.
Present. You cannot know which carbohydrates. All the carbohydrates will answer this test. So if you have a positive result, you will tell carbohydrate is present. Okay. So this is the first test you can do. Among the chemical test, the first test which you can do is the Maulish test, or it is known as alpha naphthol reaction, because the reagent has alpha naphthol, so it's also known as alpha naphthol reaction. Okay, the next test which you do. Okay, so you can do a iodine test to check whether it is a reducing sugar or a non-reducing sugar. Sorry, you do the iodine test to check whether it's a polysaccharide or monosaccharide. So I'll tell you later, but now I'll just tell you this test. In iodine test, what you do is you take the sugar solution and you will add iodine reagent to it. So when you add iodine reagent to your carbohydrate solution, two possibilities. One is the negative test will not change color. Iodine will be like light yellow to orangish color. So when you add iodine reagent, this is the color you get. So this is a negative test. But if it is a positive test, it will form a dark blue color. This is not black. You can observe it's a dark blue color. So when you do iodine test, you take the carbohydrate solution, you will add iodine reagent to it. Negative test is no color change. Positive test will be dark color change. Okay, so when will you get this test is positive only for polysaccharide. Okay, iodine test you will get positive only for polysaccharide, not for any other carbohydrates. Is it clear now to everyone? Which will
yes, ma'am.
answer the iodine test? Only polysaccharide.
Polysaccharide.
Okay, so you can distinguish whether it's a polysaccharide or not. Okay, so then what are the other tests which you can do? Benedict's test. Okay, so Benedict's test or Felling's test is what you can do. Both are same. It checks for the presence of reducing sugar. Both Benedict's test and Felling's test are done to check for the presence of reducing sugar. Whether the given carbohydrate is a reducing sugar or not, you can test it by doing Felling's or Benedict's test. So what happens in these tests is, first I'll explain the Felling's test. In case of Felling's test, you will take the sugar solution and you will add Felling's reagent to it. Two reagents, Felling A and Felling B. 2 ml of Felling A, 2 ml of Felling B, and you will add carbohydrate solutions to it and you will boil it. Okay, so when you boil it, after boiling, if you get a brown color precipitate like this, both will give you brown color precipitate. The Benedict's reagent and Felling's reagent will be blue in color, and you add sugar solution. On heating, on boiling, if it turns to brown colored precipitate containing solution, it is a positive test. If it remains blue in color, it is a negative test. So if it turns into a brown precipitate, it shows the presence of reducing sugar. So why is Benedict's or Felling's test done? To check for the presence of reducing sugar. Okay, when do you call it? It contains a reducing sugar if it has the brown precipitate. Okay, now what is happening here? Carbohydrates have been added the ability to reduce, they will reduce the copper ions present. Okay, so if the copper ions are present in both Felling's reagent and in the Benedict's reagent, copper ions are present. It can reduce the copper ions. If the reducing sugars are present, they can reduce the copper ions and they will bring the color change. And now they can act as a strong reducing agent in presence of a basic condition or an alkaline condition. Okay, so your monosaccharides can act as a reducing sugar more easily in alkaline condition. Okay, they have the property of reducing the copper ions in an alkaline medium. So this Felling's reagent will have a strong alkaline activity where your carbohydrates will form ene diols. Okay, so understood? So when the carbohydrates, if they are reducing, they can have the reducing activity in alkaline medium. Both Felling's reagent and Benedict's reagent will give you a alkaline medium. Okay, so see here, in case of Felling's reagent, KOH is present, which is providing the alkaline condition. In case of Benedict's reagent, sodium carbonate is present, which will provide the alkaline condition. But both will contain copper. Benedict's also has copper sulfate. Felling's also has copper sulfate. So what is providing the alkaline condition in case of Felling's reagent? Potassium hydroxide. In case of Benedict's reagent, sodium carbonate. So in alkaline condition, your sugars will be converted to ene-diols, Enediols. So the ene-diols will have strong reducing property. So they will reduce the copper ions. Okay, so then when they reduce the copper ions, it forms a reddish brown precipitate. Is it clear to everyone? The
Yes, ma'am.
Benedict's test. So both the tests are done to identify whether it's a reducing sugar or not, whether the carbohydrates have the ability to reduce or not. Okay, so these two tests are to check for the reducing property. When in an alkaline medium, they will be converted to ene-diol. So remember this concept shortly. In your books, there's a large information. So please try to write it down or make a note. In case of Benedict's test and Felling's test, it's just used to check whether there are reducing carbohydrates or not. So in alkaline medium, the sugars will form ene-diols, which will reduce the copper ions and form a reddish brown precipitate. Okay, so we have discussed Maulish test. Why Maulish test is done for to check for the presence of carbohydrates. Benedict's and Felling's to check whether reducing sugars are present or not. Iodine test to check if polysaccharides are present. Okay. Okay, next we'll go on for the Barthold's test. So this Barthold's test is to check whether it's a monosaccharide or a disaccharide. So Barthold test can distinguish between a monosaccharide and disaccharide. It's a reducing sugar. After doing the Benedict's and Felling's test, you want to check whether the reducing sugar is a monosaccharide or a disaccharide. You will do the Barthold's test. Within the reducing sugar, it will tell whether it's a monosaccharide or a disaccharide. Clear why Barthold's test is done? So how do you do Barthold's test? A Barthold's reagent is copper acetate in acetic acid. Okay, so Barthold reagent is containing copper ions but in acidic medium. Now in acidic medium, only monosaccharides can reduce to form a reddish brown precipitate at the bottom of the test tube. Observe Barthold's test. If they are reducing, they can form the brown precipitate only at the bottom, not like this throughout. The positive test is you will get a brown precipitate only on the bottom. If there's no change after heating, it's a negative test. If it is getting a brown precipitate only at the bottom, it's a positive test. So what is positive and negative test telling? So in acidic medium, only monosaccharides can act as reducing sugars. So whether monosaccharides will give you a positive test or disaccharides will give you a positive test. Which will give you a positive test?
Monosaccharide.
Okay. So if you're not getting, it is a disaccharide. If you're not getting a reddish brown precipitate at the bottom, it's a disaccharide. So remember that monosaccharides can act as reducing agent in acidic medium. Disaccharides cannot act as reducing agent in the acidic medium. So only monosaccharides will answer this test. So why is Barthold's test done? After Benedict's test, you identify it's a reducing sugar, and you know that monosaccharides are reducing sugar and disaccharides like maltose and lactose are reducing sugar. So now you want to identify whether it's a monosaccharide or disaccharide, do the Barthold's test. Only monosaccharide can reduce in acidic medium. So if it's a positive test, it's monosaccharide. If it's negative test, it's disaccharide. Okay, is it clear to everyone the principle of Barthold's test?
Yes, ma'am.
Okay. Now the last test which you do chemically is Salvinov's test, and this is a microscopic examination which you will be doing, Osiso test. Okay, let's discuss the Salvinov's test. Okay, so this Salvinov's test is used to distinguish between a aldose and a ketose. Okay, can you give an example for an aldose and a ketose? Which is a aldose sugar and which is a ketose sugar? First, I showed you the slide. Aldose and ketose.
Aldose can be glucose.
6 carbon atom containing sugars, which is aldose and which is ketose.
Ma'am, glucose is aldose and
Yes.
fructose is ketose.
Okay, so good. So you can use the Salvinov's test or resorcinol hydrochloric acid test to distinguish between a aldose and ketose. Generally in lab to distinguish between glucose and fructose. Okay, you can distinguish whether it's a glucose or a fructose by doing this Salvinov's test. So in this you'll use a reagent known as resorcinol in hydrochloric acid. So that's why resorcinol hydrochloric acid reaction. So you will take Salvinov's reagent, add few drops of sugar solution to it, and you will boil it, and after boiling if you get a cherry red color like this, it is a positive test. So only ketosis will answer the Salvinov's test. So this is a negative test. This is a positive test. So when you get a cherry red color, it is a positive test, indicates the presence of ketosis. Whereas aldosis will not answer this test. Aldosis will be negative for the Salvinov's test. No red color. When there's a red color, it shows the presence of ketosis. So you can distinguish between glucose and fructose by doing Salvinov's test. Okay, so have you all understood all these tests now? Whatever shown, Maulish, Benedict's, Iodine, Barthold's and Salvinov's. Is it clear to everyone now? How are these tests done?
Yes, ma'am.
Yes, ma'am.
Okay. So one more important qualitative test which you can do is the Osiso test or the phenyl hydrazine test. Here you will treat your sugar solution with this phenyl hydrazine. Okay, so when you treat your sugar samples with the phenyl hydrazine, they will form crystals. Okay, so when you treat your sugar solution with phenylhydrazine and sodium acetate, when you boil it, they will form crystals. And you will not observe any color change. Here you have to observe under a microscope. Okay, so the crystals you have to go and observe under a microscope. Under the microscope, you can differentiate which sugar it is. So you take your sugar, you add phenylhydrazine to it, sodium acetate to it, boil it until it forms a crystal. After it forms a crystal, take a small part of the crystal and observe under microscope. So when you observe under a microscope, see each carbohydrate can show a distinct shape under microscope. Glucose forms a broomstick shape structure. We call it as a broomstick shape structure. Does it appear like a broomstick here? The crystals?
Yes, ma'am.
Okay, so when it is a broomstick shaped crystal, you can identify it as glucose. When it is needle like structure like this, you can say it is a fructose. And when you get a rhombus shaped crystals, you say it is galactose. When you get a ball like structure, you say it is lactose. When you get a sunflower shaped crystals, we say it as maltose. So this is how each sugar will look when you treat it with osazone and then observe under microscope. So this is known as glucose osazone. This is galactosazone. This is fructosazone crystal, lactosazone crystal, maltosazone crystal. So you can observe under the microscope and tell what sugar it is. Is this test clear to everyone now, Osiso test?
Yes, ma'am.
Okay, so have you all understood why all the tests are done? To distinguish between what the different tests are done. Okay, now you all can take a break for just 5-10 minutes. Then I'll show you the video and very important part, how to, when you come to a practical, how to identify the test, and I'll give you certain assignments, so all these will do. Now you can take a break for 10 minutes and then come back for the class.
Yes, ma'am.
Okay, is everyone there in the meeting? Can we start?
Yes, ma'am.
Okay, I'll show you a video like how these tests will be done. You can observe that and then I'll teach you like how to systematically follow the analysis to check what is the carbohydrate given. So now I'll display the video.
For this experiment, the following materials are needed.
Is it visible to
Test
everyone?
tubes, droppers,
Yes, ma'am.
Yes.
and the following are the main reagents. Felling's A and B, Benedict's reagent, Salvinov's reagent, Maulish reagent, Barthold's reagent,
Not
and
audible,
iodine.
madam.
Not audible
Carbohydrate solutions
to
are
everyone or
glucose,
only for few persons?
fructose, lactose,
Yes, ma'am,
sucrose,
it is not audible.
starch.
Okay,
We
just a
also
minute, I'll
need
check.
test tube racks, marker, and paper tape that you can easily take note of the observations that you will
Now,
be
is it
making.
audible?
Test tubes, droppers,
No, madam.
Very low.
blender, and the
No?
following are the reagents. Felling's A and B,
Not audible?
Benedict's reagent, Salvinov's reagent,
Okay, you can
Maulish
just
reagent,
observe. I'll
Barthold's
explain it
reagent,
to you.
and iodine solution.
You can see what they're doing.
Carbohydrate solutions are glucose, fructose, lactose, sucrose, and starch. We also need test tube racks, marker, and paper tape for labeling. And before anything else, review so that you can easily take note of the observations that you will be making while watching the
Yeah?
experiment. And now that everything
Yeah, somebody who's told me anything?
No, madam.
Okay.
when transferring the carbohydrate samples. Now that everything is ready, let us now start. For the first part of the experiment, we will be doing Maulish Test, which is a general test for carbohydrates. Concentrated sulfuric acid will also be used in this test. Make sure that you also label your test tubes so that you won't get confused when transferring the carbohydrate samples. First, we need about 2 ml of each carbohydrate solution in our 4 test tubes. And then we will be adding 10 drops of Maulish reagent to each carbohydrate sample and then mix thoroughly. And then carefully add about 15 to 20 drops of concentrated sulfuric acid and allow it to flow down the side of the tube. Do not stir. The sulfuric acid will form a bottom layer, and then note the color formed at the zone in between the two liquids. And here are the results after the addition of concentrated sulfuric acid. Here are the close-up videos for each of the tubes. Our next test is Benedict's test, a test for reducing sugars. To do this, about 2 ml of Benedict solution is added to 4 empty tubes and then placed into a water bath for 30 seconds. the solution remains clear blue, we can continue with the test, which means the reagent is not contaminated. Allow the 4 test tubes to cool down until they are just warm to touch, and then add 5 drops of the carbohydrate solutions. After that, place the 4 test tubes containing Benedict solution and carbohydrate samples into a boiling water bath for 2 minutes. Cool and observe what happens. Always remember to take note of your observations by writing it down so you won't have to rewatch this experiment when answering your laboratory worksheets. Here are the samples just after taking it out from the boiling water bath. We have allowed it to cool down for a few minutes, and here are the results. Our next test is Felling's test, which is a reduction test to determine the presence of reducing sugars. It differs from Benedict's test in that Felling's reagent contains Rochelle's salt or sodium potassium tartrate in place of sodium citrate. Felling's solution consists of solution A and solution B. So for the procedures, in each of the test tubes, we are going to mix 1 ml of Felling solution A and 1 ml of Felling solution B and add 3 ml of water. After that, the 4 test tubes are placed in a boiling water bath for 1 minute. If the solution remains blue or clear blue, we are going to add 8 drops of each of the sugar solution to each of the test tubes, and then boil again the 4 test tubes with the sugar solution for another 2 minutes and note the changes in the solution. And these are the results after 2 minutes in the boiling water bath. Here are the close-up results. Our next test is Barthold's test, which distinguishes reducing monosaccharides from reducing disaccharides by controlling pH and time of heating. This is also a copper reduction test in acidic conditions. For the procedures, in each of the 4 test tubes, we are going to add 2 ml of Barthold's reagent. And then, to each of the test tubes, we are going to add 10 drops of each of our sugar solution. And then, place the test tubes in boiling water bath for 5 minutes. After 5 minutes, we are removing the test tubes from our boiling water bath and let it cool. Observe the first 15 minutes. Record your observations. Our last test is Salvinov's test. This test distinguishes between fructose and glucose. Overheating of the solution should be avoided because on continuous boiling, aldose test will also give a positive result. To do this test, we're going to add 2 ml of the sugar solutions to 3 ml of Salvinov's reagent. After that, immerse the solutions in boiling water bath. Observe the color changes during the first 10 minutes of boiling. And here are the results after taking out the test tubes in the boiling water bath. And now we will be proceeding with the iodine test for starch. For this test, we are going to place 2 ml of the plant starch solution and then add a drop of iodine solution. And then heat the solution and note the change. After heating the solution and cooling it down, here is the final result for the iodine test. And that's it. See you in our next experiment.
So did you watch how the different tests are done? Only thing is, for the Benedicts and for the fillings, we usually mix the solution. I'll show you another video, you can observe that. In this Maulish test, you saw how the ring appears. Then Salvinov's test, you would have seen how the red precipitate is got. Barthold's is right only at the bottom, but only difference, the Benedicts and fillings, we usually mix it when we boil. In this, they didn't mix. I'll show you another video. You can just watch that.
Looking at carbohydrates. So different types of sugars, and we will be seeing some of their different chemical reactions. We're skipping part A, so not looking at any food samples, so we're going to jump right into part B, which is Salvinov's test for ketoses. You might want to pause it a second here. This tells you which test tubes 1, 2, 3, 4, and 5 are all day. So again, Salvinov's test. We're just going to follow the procedure here for part B. So first we're going to add 1 milliliter of sugar solution to each test tube. So that was glucose going into the first test tube. Going into the second test tube is fructose, fruit sugar. Going into the third test tube will be sucrose, which is table sugar, just dissolved up in water. All of these are sugars dissolved in water. The fourth sugar is maltose. And the final sugar is a polysaccharide, starch. And you can see starch doesn't fully dissolve, so the solution was a little bit cloudy on that. A, B, C, D, and E are the 5 unknowns you'll be figuring out today, so we don't know what sugars they are, but I've already filled those up. So you're going to be looking for a comparison between those 5 unknowns and the known sugars all day long. We're going to add Salvinov's reagent. It has a strong acid in it, so we would want to be careful with this in lab if you were doing this. We add 1 milliliter of this reagent to each test tube. And then we've already got a hot water bath going, and so we're going to heat these. Heat speeds up reactions. So we're going to heat our unknowns, and we're going to heat our knowns in some boiling water. You want to watch for a color change in these. A dark orange or red color would be a positive test for ketoses. And we will heat these for 5 minutes, so you might see some color change over the course of 5 minutes. Note that I've edited this to make it take less than 5 minutes. But you can start to see a bit of yellow, a bit of pink start to darken here as time is going. Getting even darker. And the bubbles there are outside of the test tubes. That's just the boiling water. So we heat it, and you'll see these in a minute, but that A came out pretty dark red, B, not much color, C, got some color, D, not much happening there. If we heat these long enough, they would all react and start to turn red, so that's why we don't. So you can see the results there, lots of color in A and C, right? And we're hoping that will match some of our known sugars here, 1 through 5. So I'm going to carefully take those out. So there's 1, not much color. 2, dark red color. Remember that dark red is a positive test for ketoses, so there must be a ketone in that sugar. 3, positive. 4, not seeing much. And 5. So that was Salvinov's test for ketoses. We want to test lots of different aspects of these sugars. Here again were those unknown results for the ketoses. So now we're going to do Benedict's test for reducing sugars. Reducing sugars are sugars that can be oxidized. And so these are going to be sugars that usually have a free aldehyde that can be oxidized is most often what you'll see. Adding the same sugars, so sucrose, again in number 3. 4 will be maltose. Maltose is just 2 glucoses connected to each other, and so when we break down starches, you get a little bit of malt flavor because of the maltose sugar. So I've got my known sugars here. We're going to add Benedict solution to these now. Benedict solution, we'll add 1 ml of each of those. Notice that that blue color is just part of it. Here's my unknowns. Again, same A, B, C, D, E sugars that I grabbed. The blue is not a positive test. That's just because there's some copper in the Benedict solution. So I have my same boiling water bath ready. Again, I'm going to give each of these some heat for 5 minutes and watch how they change as the reaction goes. So this time I'm looking for a reddish solid to form. If there's a lot of sugar, if there's less sugar, I'll see some green and yellow kind of solids form. Again, this is not true to time, it was edited, but you can start to see a little bit of yellow solid forming in some of those test tubes, and we'll take a closer look at them when they're done with this heating period. The ones that are stained blue and transparent, that would be no reaction. You can see now the yellow solids are becoming orange in some of the test tubes. You can tell there's a solid there because it's opaque, meaning you can't see through it. So solids, even if you can't see the solid at the bottom, if you can't see through the test tube, that means there's a solid
usually
there.
mix it like this
And
to
you
get
can
the
see like those orange ones, those look pretty similar to each other, right? And that's what we're looking for, is something that looks the same between the known and the unknown. Color's getting darker with a little bit more heat. Getting ready to take these out pretty soon. This is just a fun one to watch. It's cool colors to see. And on the first page of your experiment, you have some test results that you can compare these to, the different colors, and what percent of glucose they correspond to. So test tube A, not much color there. Test tube B, we'll get a closer look in a minute, just a touch. That would probably not be a positive test. Here's 1 through 5, so you might want to pause that so you can get a closer look at it. And then here's A through E. Those 2 on the left, I would say, are not positive, or B, you might say, just has a very slight amount of glucose in it, or not glucose, but reducing sugar, sorry. On to part D now. We're doing Barthold's test for monosaccharides. So these are going to be sugars that just have 1 ring, right? That's what a monosaccharide is. So we're going to add those same sugars, fructose in test tube 2, sucrose in test tube 3, maltose coming in test tube 4, and then we'll have our cloudy starch solution for test tube 5. So for Barthold's, we've added those sugars. Now we're going to add Barthold's solution. Again, this has some copper in it, and that's why it's a blue solution. The blue doesn't mean there's been a reaction. It just means that we have the original Barthold's reagent. So there's 1 through 5. I've prepared A through E, the unknowns, off screen for you. And so now I'm going to be ready to heat these for 2 to 3 minutes. Got a little different angle on the heating this time. Cinematography is just amazing on these videos. I think you'll have to agree with me. This was done in such a time crunch. So we're going to watch those heat for a few minutes here. Again, it's been edited to speed it up, but we're looking for a dark red precipitate or solid on the bottom. That would be a positive test for a monosaccharide. Not seeing much here initially, but keep your eyes on it. You can see some of those blues starting to sort of change color, and those are probably going to be our reactions, like 1 and 2 look a bit reactive there. Again, the heat helps molecules move faster, so the heat speeds up these reactions, and we need that help on all of these. Now you can start to see some more red showing up. Here's your final results, 1 through 5. 1 and 2 definitely have some red solid in them. 3, 4, and 5 are looking nice and blue and transparent. Here is A through E, and again you can see in C and in E some dark red solid there at the bottom. The others we don't see any red solid in, so blue means nothing changed. So one last round, we've cleaned the test tubes out. We're going to do the iodine test for starch now. This one you do not heat, it's just going to be a quick instantaneous reaction. So we're going to put our sugars in, glucose again, and I believe it was fructose number 2, sucrose in 3, maltose coming in 4, and starch coming in to test tube 5. So on this, we're going to add just a couple drops of iodine solution. This is the same iodine you would have at home. When I add a few drops, iodine is already normally yellow, so that yellowish tinge doesn't mean anything. That's a negative test for polysaccharides. Keep adding to each of these. And here you see something entirely different. That is a very vivid positive test for a polysaccharide. It's like a deep purple, blue kind of color. These others, even if I mix them around, don't turn that deep purple color. Here's the same test on my unknowns. So we're looking again for that purple color means I have a polysaccharide present. So not seeing a polysaccharide there. Very obvious purple. Again, the yellow is just the color of the iodine. And so this is some really solid data for you, right? You should be able to connect one of your unknowns to your knowns.
So now basically we saw how all the tests are done. Okay, that was to show you how we add the sugar solution, how we add the reagent. So if you had observed, you saw everything was with just a dropper. Since it's a qualitative test, you need not add it very accurately, right? So this, you take a test tube, just add few drops like that, few drops of sugar, reagents. You will boil it. This is how you will do the test. Now all of you listen carefully. I'll show you step by step how you can analyze a given sugar, and I'll give you an assignment based on that. So it's not theoretical part. You have to understand very carefully now. So listen carefully. So what I'm teaching now. So we have given you an unknown solution. How will you start doing the test? So which is the test which is general for all the carbohydrates?
Maulish test.
Okay, so first to say whether carbohydrate is present or no, you do the Maulish test. Okay, what is the positive result for Maulish test? What is
A
the positive result?
ring will form, ma'am. A violet ring.
Violet or brown ring. So positive carbohydrate is present. Now you have your aim is to find out which carbohydrate. So first do Maulish test. Ring is formed, confirms the presence of carbohydrates. Okay, next which test you will do is you have to do Benedict's or Felling's test. Any one you can do or you can do both. Okay, what is it used to distinguish between Benedict's and Felling's test? It's used to distinguish
Reducing sugar.
reducing and non-reducing sugars. Okay, so Benedict's and Felling's test you do. If you get the positive test, what does it say? Presence of?
Reducing sugar.
Okay, if you get a negative test,
Non-reducing
Non-reducing.
sugar.
non-reducing sugar or presence, absence of reducing sugar. Okay, now we have to divide based on the result. If you are getting a negative test, what are the non-reducing sugars? Monosaccharides, are they non-reducing? No.
Disaccharide.
In disaccharide, which is non-reducing? In disaccharide, which is non-reducing?
Sucrose.
Sucrose. And
Sucrose.
polysaccharides, are they non-reducing?
Yes, ma'am. Polysaccharides are
Okay.
non.
Yes. So,
Completely
non.
non-red.
So, if your Benedict's test is negative, there are two options. Either it should be a polysaccharide or it should be a sucrose. Yes or no? If your Benedict's test
Yes,
is
ma'am.
negative, it's a non-reducing. Non-reducing, only two options. It should be either a polysaccharide or a sucrose. Okay. So, if you get a negative test, you will go and do the iodine test. Okay, if your Benedict's test is negative, what you should do next is directly go to iodine test. You need not do Barthold's test, Salvinov's test, which is further to distinguish between reducing sugars. So if your Benedict's test is negative, you will start doing the iodine test. Now, what is the positive result for iodine test? What color will develop?
Dark blue.
Deep blue, purplish.
Yeah.
Dark
Which
blue.
Deep
one
blue.
will give you blue color? Which is the only carbohydrate which will give you a dark blue color? Which group of carbohydrates?
Polysaccharide.
Polysaccharide.
Polysaccharides.
Yeah.
Star.
So do iodine test. If it is positive, it is polysaccharide starch. If it is negative, it is sucrose, which is a non-reducing sugar. So this is how you can identify a starch or a sucrose. So Maulish test first generally. If Benedict's test is negative, go for iodine test. If iodine test is positive, it is starch. If it is negative, sucrose. Okay, with this three tests, you can identify whether it is starch or sucrose. All these tests are not applicable for your starch and sucrose. You need not do these tests. If your Benedict's is negative, you have to go directly to the iodine test. Okay. In case you are getting Benedict's test positive, what does it show? It is a reducing sugar. Either it can be a monosaccharide or a disaccharide. Yes or no? What is the test which you can distinguish between monosaccharide and a disaccharide?
Barthold's test.
Okay, Barthold's test positive confirms,
Mono
Monosaccharide.
negative will show it is.
A disaccharide.
Why positive will show, why monosaccharides will show a positive test in Barthold's test? What is the
Because
principle?
they in acidic medium only monosaccharides
Very
can
good. They can
reduce.
reduce in acidic medium monosaccharides. So first, when Benedict's and Fellings is positive, you will move on to Barthold's and say whether it's a monosaccharide or a disaccharide. Okay, when it is positive, it's a monosaccharide. When it is negative, it's a disaccharide. Okay, so for positive, what will you do? You know it's a monosaccharide, but you don't know whether it is a aldose or a ketose. But you know it's a monosaccharide. To distinguish between aldose or a ketose, you will go and do Salvinov's test. What is the color change for Salvinov's test?
Cherry
What is the
red.
cherry red? Which will give you a cherry red color?
Fructose.
Fructose. Ketosis will give. So when Salvinov's is positive, it is fructose. When Salvinov's is negative, it can be glucose or galactose. All these are aldoses, but how will you further distinguish? You have to do Osiso test. Observe under microscope. Observe the crystals. Difference between glucose and galactose. Then confirm whether it's glucose or galactose. So do the Osiso test. Okay, so this is one branch. Okay, when Barthold's test was negative, you know it's a disaccharide. So what are the two disaccharides which are reducing sugar?
Maltose and
Lactose.
Maltose and lactose. So how can you identify them? Because sucrose is coming here under non-reducing. So if Barthold's test is negative, you have two disaccharides. Either it can be a maltose or a lactose. So what you should do, you have to do a Osiso test and observe the shape of the crystals. If you get a sunflower shaped crystals like this, maltose. If you get a ball shaped crystals like this, lactose. Okay, so this is how you will do it in a lab systematically. You will not do all the test for all the unknown given, you have to start. Okay, I've given you an unknown solution. Take that unknown solution, do Maulish test first. Then do Benedict's test. So Benedict's will decide whether you have to do a iodine test or a Barthold's test. Okay, when will you do a iodine test? Which Benedict's test? When Benedict's test is negative or positive?
Negative.
Okay,
Negative.
when it is
Non-reducing sugar.
negative,
Negative.
do iodine. Okay. I think you have all understood this flow chart. So now when you get a starch or a sucrose, you will diverge from here. You need not do Barthold's test, Salvinov's test, Osiso test, all these are not required. So the data sheet for starch and sucrose will first have Maulish test, followed by Benedict's test, iodine test, then the what is the inference and what is the result. Okay, so you will start writing like this for each sugar. Sample 1, solubility test, Maulish test. You continue. After Maulish test, Benedict's test. After Benedict's test, iodine test. What is the observation? Observation you will write, a brown ring was observed or a reddish brown precipitate was observed. Okay, in case of iodine test, a deep blue color was seen. Okay, here if it is negative, you can tell absence of reddish brown precipitate, absence of blue color. Okay, then inference you write confirms the presence of reducing sugar or confirms the presence of aldose, confirms the presence of monosaccharides or disaccharide. So for each test, I've given you here. So do the test. Then in the inference you will write whether reddish brown precipitate, presence of reddish brown precipitate, absence of reddish brown precipitate. In the second column, observation you will write as presence of, for Benedict's test you will write the presence of reddish brown precipitate. And what will be the inference? Confirms the presence of reducing sugar. Okay, for Maulish test, what will you write here in the observation? If it is a positive result,
violet or brown ring.
presence of
Carbohydrates.
a violet or brown ring. What will be the inference?
Confirms presence of
Confirms
carbohydrates.
presence of carbohydrates. Like this, you have
Carbohydrates.
to write for each test, and I'll give you 2 sugar samples for which you have to write it. Okay, later, I'll do that later. Now we'll do one work here. So now can you tell me, you have done a Maulish test, this is positive, Benedict's test is negative. So what will you do now next if Benedict's test is negative?
Iodine test.
Okay, iodine test.
Iodine test. We go with the
You
iodine
have
test.
done iodine test and iodine test is positive. Okay, now tell me what will this carbohydrate be?
Starch or sucrose?
Starch or
Starch.
sucrose?
Starch. Iodine test is positive. So will it be starch or sucrose?
It is starch, ma'am. If it is
Starch.
positive.
Fructose will not show positive iodine test. Understand very clearly. Iodine test you do when Benedict's test is negative. Either it can be a starch or a sucrose. But how can you distinguish between a starch and sucrose? Do iodine test. That blue color you will get only for starch. If you don't get blue color, it is sucrose. Understood?
Yes,
So
ma'am.
what is this carbohydrate here
Yes,
now?
madam.
First one.
Starch.
Okay.
Starch, ma'am.
So please make a note of it. You have to fill this and submit for the assignment. Okay, now sucrose. Will the Maulish test be positive or negative?
It is positive, ma'am.
Positive.
Positive.
Benedict's test will be positive or negative
It is negative.
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 because it's a non-reducing sugar. So will you do Barthold's test and Salvinov's test?
Ma'am, we
No,
No.
cannot.
ma'am.
Okay,
No,
you
madam.
write it as Na, and then you do iodine test. Will it be positive or negative?
Negative, ma'am.
Negative.
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 Okay, so understood how to fill the table now? Okay, now one more thing. We'll see who finds this out. Tell me what is this third carbohydrate? Maulish positive, Benedict's positive, Barthold's positive, but Selvinov's negative.
Um,
Ma'am, glucose or
So,
galactose?
Glucose or galactose, very good. Okay, it's going to be either glucose or galactose. You can do Osiso test and find out further. Okay, now for fructose. Tell me Maulish will be positive or negative?
positive, ma'am.
positive.
Positive, ma'am.
Good. Benedict's,
Positive.
Positive.
Barford's,
Positive.
because
Positive.
it's a monosaccharide, positive. Selvinov's,
Positive, ma'am.
positive because
Positive.
aldose. Iodine you're not doing. Iodine will be only if Benedict's is negative. So write it as Na here. When you're not doing the test, you have to write it as not applicable, Na. So fructose. Okay, now come on, lactose we'll see. Maulish test positive or negative?
Positive.
Positive.
Benedict's,
Positive, ma'am.
Positive.
Barfords,
Positive, ma'am.
it's a disaccharide. Barford will be
No precipitate.
Negative.
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 No
Okay, you
precipitation.
remember. Barford test will be negative for disaccharide. Will you go do Selvinovs and iodine for that after finding out it's a
No,
disaccharide?
ma'am.
What will you
No,
do?
madam.
Ozone test.
Ozone test.
Ozone
So
test.
Selvinov and iodine is Na, not applicable. Understood? No, all of you should do this now and then you will upload the assignment. So I'll give you few more assignment. So you start writing this, take a picture of it, start writing this. This is the first assignment and the other assignments I'll give you shortly. Just take a picture of this, start writing and fill in the blanks. I'll give you the questions for the other assignment. Have you all, please take a screenshot all of you. Then start writing the answers for this.
Are you okay?
Sorry? So fill in the blanks, which is positive, which is not applicable, which is the sugar. So I do that. Okay, have you all taken the screenshot of it?
Yes, ma'am.
Yes,
Yes,
ma'am.
madam.
Okay, so I'll show you the remaining questions. Keep doing this. I'll show you the other questions for the assignment. So if you are finished with that assignment, the next assignment is you have to briefly give the principle for Benedict's test, Felling's test, Barthold's test, Salvinov's test and Iodine test. Okay, you can just see the PPT. Okay, so the principle I've written there I've explained also. What is the principle? Please write down the principle from here for all the tests. It's there in your books also. I don't want the structure and all that, only that principle which is given here. Okay, so you can write down the principle for these following tests. Yeah?
Yes, ma'am.
Anybody had any doubts?
No, ma'am.
So after this table, kindly write the principle of all these tests. Have you finished this? Can I show you the next assignment?
Ma'am, we are still writing, but you can give the next assignment. We can finish together.
Yeah, I'll just explain. You can just see. Next assignment is you have to write the test, observation and inference in the form of the tabular column like this. Okay, you write sample 1 on the top and write here sample 1, and then you have to draw tabular columns, test, observation and inference. Okay, then you have to write for these two carbohydrates what are the tests which you will do. For example, I explain now. You have to write the solubility test. Okay, in simple terms you write. Though it is shown here in this PPT, all so much of explanation is not needed. Methodology they've given, right? So you can write. Take 2 ml of distilled water to a test tube. Okay, and cold water and add sugar to it, dissolve it. Okay, like that simple words you write. Okay, put a little of carbohydrate sample. You will not mention glucose here because I am giving you unknown carbohydrates. So you will write it accordingly. You will tell, take few amount of the carbohydrate sample given, add distilled water to it, dissolve it. Okay, that is the methodology. In the test, you will be writing that. So the solubility test, you will explain how to do that test in simple terms, then write the observation. The sugar was completely soluble or sugar did not dissolve. Okay, it had turbidity. Like that you can write whatever is the observation. Write the inference. If it is completely soluble, you can say shows the presence of monosaccharides and disaccharides. If it is turbid, shows the presence of polysaccharides. Understood what I'm trying to say?
Yes, ma'am.
Okay, then do the next test. Maulish test obviously will be positive for everyone. Write how to do the Maulish test here. So as I told you, take the sugar solution, add 2-3 drops of Maulish reagent, mix it. Okay, add H2SO4 drop by drop to the walls of the test tube. Like that, simple you explain here and observation. Obviously, you'll get brown ring. Okay, you write appearance of brown ring or violet ring at the junction, okay, or appearance of the brown ring, you can just stop that, or violet ring you write, and then write the inference. Confirms the presence of carbohydrates. Okay, it shows only the carbohydrates. Okay, what will be the next test which you will do for any sugar?
Benedict Solution.
Write Benedict's test. Don't write both Benedict's or Fellings. Write any one of the test. Enough. Okay, then you write third test, Benedict's test. How will you do it? Take Benedict's reagent, add few drops of sugar, carbohydrate solution to it. Keep it in boiling water bath for 2 minutes or 3 minutes. Okay, what will be the observation if it is starch? Will you get a positive result here?
It will get negative, ma'am.
For Benedict's test, will you get a
Negative.
positive? Negative. So you will write
Negative.
absence of reddish brown precipitate. Okay, for starch, Benedict's test you will write as absence of reddish brown precipitate. But fructose, what will be the inference?
Presence of
Yeah, presence of reddish brown precipitate will be the observation. Inference you will tell, if it is positive, you will tell confirms the presence of reducing sugar. If it is negative, absence of reducing sugar. So you can use this which is given here. So if it's positive, you write presence of reducing sugar, absence, all that you can write. Then for starch, what test will you do after Benedict's test?
iodine.
Iodine test, right?
Iodine test.
Yeah, go for directly iodine test. So for starch will contain solubility test, Maulish test, Benedict's test and iodine test. You don't have to write Barthold, Salvinov's, all those tests. Understood what you should do now? For
Yes,
starch,
ma'am.
you will write Then for fructose, what will you do? You have to do solubility test, Maulish test, Benedict test,
Benedict test,
after
Barthold
Benedict
test,
test,
Barthold,
Barthold test,
Barthold,
then
check whether
Salvinov test.
monosaccharides or disaccharide, then do Salvinov's test, positive or negative, like that. You have to write for these two sugars. Draw the tabular column like this, and then write the reagent test. Okay, how you do the test in simple words, what is the observation, what is the inference. And at the end of the table, you write the given carbohydrate sample was found to be. Since I told you here already, because you're not doing it practically, okay, you write it as on the top, you write it as sample 1, and result you write the given carbohydrate sample was found to be starch. And for second, you write sample 2, do the test, write all the observation result, and final result you write result, the given carbohydrate sample was found to be fructose. Understood about this assignment?
Yes, ma'am.
Okay, so if you want, you can take a screenshot of all the assignments once again. I'll just display this. I hope you should have done it. This is the next assignment. Just the principle for each. Then this is the assignment. I've explained you how to do it. So please draw the tabular column like this and do for these two sugars. So I'll share the Google form links now. Once you complete, you can upload it. After you complete the assignment, you can upload it. I'll just share the Google form link now. Okay, can I stop sharing the PPT? Have you all written down the questions?
Yes, ma'am.
Okay. Yes, ma'am.
Okay. I'll stop sharing the PPT and share the Google form links for you. I've shared the Google form link. Now in your WhatsApp group also I will share the link and also the videos. You can take your time, write the assignment and submit it by today evening. You can take time till today evening. Is it fine to all of you?
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Okay, the meeting will be on. Okay, any doubts you can ask me. You can take time till evening to submit the assignment. You can take your time. Okay, I'll share in the group the videos and then also the Google form link. I'll share it to you once.